Yeah, yeah, just now. Yeah, yeah.
Hello and welcome to Chai and Why. This is a very special month, November, where we have sessions on every Sunday celebrating the Nobel Prizes for the year. We're going to start off with the Nobel Prize in Physiology and Medicine. And this will be given, traditionally, all these talks are given by our graduate students. And we have Diksha Kumari from the Department of Biological Sciences. She is an integrated PhD student working on intracellular transport. And before she came here, she did her bachelor's at the MS University Baroda and then joined us for an integrated PhD. So over to you, Diksha, and tell us all about the Hepatitis C virus and the Nobel Prizes of the year.
Yeah, thank you, Arnab. Am I audible? Okay, I hope I'm audible. I will just start.
Go ahead, Diksha, it's all yours.
Okay. So good morning, everybody, and I welcome you from my side in today's session of Chai and Why, which is the very first session of this year's Nobel Prize talk series. The title of today's session is A Battle Against Hepatitis C Virus, which is also the title for this year's Nobel Prize
Diksha,
in Physiology.
Diksha, you are audible.
Yeah, thank you. So, which is also something which has backed this year's Nobel Prize in Physiology and Medicine, and it is awarded to these three gentlemen: Harvey J. Alter, Michael Houghton, and Charles M. Rice, as the citation says, for the discovery of hepatitis C virus. Now, before we actually go on to discuss their discoveries and why it was important and why were these people awarded the Nobel Prize, let us first understand these two keywords, virus and hepatitis. So this is a schematic representation of a typical human cell. We are composed of a type of a cell which is very nicely surrounded by a boundary, as you can see here. And inside our cell, we have very specialized organelles to perform their specific function. For example, mitochondria is there to synthesize energy. Other organelles like endoplasmic reticulum or Golgi apparatus, they are there to synthesize some important biomolecules for the cell, for example, protein and lipids, and they package these biomolecules and deliver it to their specific destinations. The nucleus, which is there at the kind of center of the cell, it encloses the genetic material, which is DNA. Our DNA is what we inherit from our parents and pass on to our next generation. And it contains all the information inside the cell to make up what we are. Now, talking about DNA, our DNA is having 3.1 billion nucleotides. To give you a sense of how big this number is, I have written it in different formats. Okay, so what is a nucleotide? To understand it, you can imagine these beads on a string model. The way a single bead is kind of a structural unit for this string, similarly, a single nucleotide is a structural unit for our DNA, and a series of nucleotides arranged one by one aside to each other forms our DNA. And as I just said, we have 3.1 billion such nucleotides in our single DNA molecule, which contains the information for the synthesis of approximately 22,000 proteins. Proteins, along with other biomolecules, are known to perform each and every function that your cell does, be that digestion of your food, your DNA replication, cell division, delivery of anything from one place inside the cell to another place, and so on and so forth. A bacterial cell, on other hand, is a simpler structure. It has a boundary which is different than our cell boundary, but inside the bacteria, there are not as many organelles as we have, as you can see from this representation also. And a bacteria, unlike us, does not have a properly defined nucleus. The genetic material of the bacteria is lying freely, kind of freely floating inside the cell in a specialized lesion called nucleoid. Okay, these terms are not important, but anyway. So E. coli bacteria, for example, which is present inside our gut, is having 4.6 million nucleotides, which is 1,000-fold less than the number of nucleotides than a human DNA. And it encodes for approximately 4,000 different proteins. This is a diagram of an E. coli bacteria, which I just said. A virus, on the contrary, is not even a true cell. It is a acellular structure composed of a genetic material, which is either DNA or sometimes another biomolecule of the same family known as RNA, and proteins or lipid molecules enclosing this genetic material. It is not able to perform the very basic functions that a cell does, for example, replication. It requires a host organism or a host cell to multiply or to produce more of its own copies. And that is why and so because it's not even a true cell, it is obviously not able to perform some other function. For example, take nutrients, metabolize nutrients, synthesize biomolecules without infecting any other cell. Hepatitis C virus, which is the focus of today's talk, is having 9,600 nucleotides, which encode for just 10 proteins. Compare it with the number of proteins that we have in our cell, approximately 22,000. SARS coronavirus 2, which has caused the current ongoing outbreak of coronavirus, so-called COVID-19, is also somewhat similar. It is having 30,000 nucleotide, coding for 29 proteins. To give you a sense of the size of a virus as compared to a human cell, I don't know if you can see this tiny line. It is a 1 millimeter length, which is probably the shortest unit of length that we can see with our naked eyes. A nanometer is 1,000,000 times shorter than 1 millimeter. And our red blood cells are having approximately 7,000 nanometer diameter. Now, mind it, red blood cells are not the largest cell, obviously not the largest cell in a human body. There are much, much bigger cells than RBCs. And viruses, on the other hand, they have a size of few hundreds of nanometers and sometimes even less than that. For example, influenza virus, one of the example of influenza virus is H1N1, which caused swine flu outbreak, is approximately 85 nanometer in diameter. Polio virus causing polio is just 27 nanometer in diameter. Similarly, HCV virus is approximately within the same range, 50 to 80 nanometer in diameter. So if this is a human cell and this is a bacterial cell, this tiny particle is a virus which infect either a bacteria or a human cell. Now, throughout our life, we must have heard of different viral diseases at one or another point in our life period. For example, some of the examples are chickenpox. It is caused by varicella zoster virus, which infect a kind of your nerve cells. Common cold, it is caused by different families of viruses. For example, rhinoviruses, which is the most common family of viruses for common cold, and coronaviruses. Polio, which is caused by poliovirus, it attacks your spinal cord, often leading to paralysis in severe cases. AIDS, which is caused by human immunodeficiency virus, it attacks your immune system. Dengue, caused by dengue virus, it attacks some specialized cells in your skin known as Langerhans cells, and in some severe cases, it can attack some other blood cells. Influenza, it is having symptoms similar to common cold, just that the symptoms are somewhat more drastic, and in severe cases, it can often lead to shortness of breath. H1N1 in swine flu virus is an example, is a very popular example of influenza causing virus. And hepatitis, which is liver infection. Let us have a look at our liver for a while. It is a very versatile organ present below our diaphragm and right above the intestine. It is known to perform a myriad of functions. Some of them are secretion of bile juices, which are important for digestion, secretion of some blood proteins and hormones. It acts as a storage reservoir for glucose. Now, what does it mean? Whenever we have excess of glucose in our blood, it is stored in a form of storage molecule known as glycogen in our liver, so that when your body lacks glucose, for example, in cases of fasting, you can go back to this storage of glucose. You can break down glycogen molecule into glucose molecules to increase your glucose levels in the blood. Another function of liver is it metabolizes some complex compounds, for example, some drugs. And this is why alcohol targets your liver primarily, because alcohol is metabolized in liver, and alcohol metabolism in liver leads to the formation of some molecules which, if present in excess, are harmful for the cell. And I don't know if we know the fact that liver is the largest internal organ present inside our body, and it is the second largest organ altogether after skin. Some symptoms of liver dysfunction are excessive fatigue. You will feel tired more frequently. Dark colored urine and stool, which is because of excess of a protein called bilirubin in cases of liver dysfunction. Yellowing of skin and eyes, which is again because of the same reason, excess of bilirubin. Swelling in abdomen and legs, which is because of lack of a protein which is synthesized by liver called albumin. And loss of weight. Simply because you are not able to digest your food properly, you may end up losing your weight. Now coming to hepatitis. It is composed of two Greek words meaning liver inflammation. So whenever there is a damage in your liver, for example, by any viral infection, or when your immune system is little bit faulty and it wrongly attacks the organs of your own body, or more precisely, it wrongly attacks your liver, it can cause hepatitis. And overconsumption of alcohol can also cause hepatitis. So what happens is that whenever there is a damage in your liver cells, neighboring immune cells come to heal this damage. And this is what is known as liver inflammation. Whenever immune cells are coming to a place to do some repair work, it often leads to inflammation. But in severe cases, what can happen is that if the damage persists for a long period, these cells fail to repair the damage. Rather, they then deposit some dead material in form of some proteins or in form of some dead cell themselves, which can lead to the formation of a scar tissue, as you can see here, rendering your liver unable to perform its function. This phenomena is known as liver cirrhosis. There are 5 different kind of viruses that can cause hepatitis. Hepatitis A, B, C, D, and E. Hepatitis A virus, HAV, causes acute hepatitis, often known as jaundice. And the most prominent symptom of jaundice is, which you can see visually, is yellowing of your eyes and skin. It is known as acute hepatitis because in most of the cases, the infection does not last for long. You clear off your viral infection within 2 to 3 weeks on your own. It is transmitted through contaminated food and water. So as a precaution to prevent the transmission of hepatitis, it is said that you should follow proper hygiene practices. For example, you should wash your hands properly after going to washroom, because an infected person will be having HAV particles in his or her stool. You should wash your hands properly before eating food. Consume clean water, which is coming from a trusted water supply, or filter your water properly before drinking it. Okay. HAB, hepatitis B, and hepatitis C virus, on the other hand, are transmitted through blood and other body fluids, and they often lead to chronic hepatitis. So this is how your hepatitis can progress if it lasts for a long period, for example, 10 to 30 years. Chronic hepatitis will lead to liver cirrhosis, which can eventually lead to liver cancer. The biggest problem in case of HBV or HCV infection is that, unlike HIV infection, you don't get any symptoms until late stage. That is, until the stage when your liver is already damaged enough. And so because you don't know that you are carrying this infection, you may go on to transmit it to other healthy people. This is how you can transmit, or this is how a healthy person can catch HBV or HCV infection, through infected needles and syringes. A pregnant mother can transmit HBV or HCV to her baby during her pregnancy. It can be transmitted through sexual practices that lead to exposure to blood. And again, following some unhygienic practices, for example, using shared razors or shared nail cutters, shared toothbrushes, etc. All of these can lead to transmission of HBV or HCV. In somewhat around 1950s or 1960s, a lot of people who were undergoing blood transfusion were reporting these hepatitis cases, and it was not known at that time what is causing hepatitis. So this is where Baruch Samuel Bloomberg, he contributed for discovering for the first time hepatitis B virus as the infectious agent which can cause hepatitis and which is transmitted through blood. Not only he discovered this virus, but he also developed diagnostic test to diagnose the presence of this virus in the patients who were going to donate their blood, so that an infected person will not donate his blood into a healthy person, and the virus infection can be prevented at that stage. And also he contributed significantly for the development of vaccine against hepatitis B virus. For his discoveries and his contribution in the field, he was awarded Nobel Prize in Physiology and Medicine in 1976. And in fact, World Hepatitis Day is celebrated on the birth date of Baruch Samuel Bloomberg, which is 28 July. This is a poster which was released on this year's World Hepatitis Day, where it is reported that 325 million people globally are having viral HB or HC infection. And an estimated 71 million people are having, out of these 325, approximately 71 million people are suffering from chronic hepatitis C infection. Approximately 900,000 deaths occur because of hepatitis B virus infection annually per year. So if you see, coronavirus over the last 6-7 months has caused approximately or somewhat more than 1 million deaths, but hepatitis B virus infection has been causing these approximately 1 million deaths per year since the time it has been discovered, that is since 1970s. This is the latest data that WHO has updated. In 2006 alone, approximately 400,000 deaths occurred because of hepatitis C virus infection. Another major concern is that 19% of people living with hepatitis C infection don't even know that they're having the infection, as I just described, that because this virus does not manifest any symptom until the late stage of disease, people don't know that they are having the infection, which highlights the problem of proper and timely diagnosis of HBV or HCV infection. Now coming on to this year's Nobel Prize, as I mentioned in the very beginning slide also, Harvey J. Alter, Michael Houghton, and Charles M. Rice bagged this year's Nobel Prize in Medicine for the discovery of hepatitis C virus. Harvey J. Alter is a US-born scientist, born in New York. He attended University of Rochester for his Bachelor's of Arts degree, followed by a degree in medicines. Then he joined National Institute of Health in 1969 as a senior investigator, which is where he conducted his prize-winning work. I would say Harvey Alter was very privileged enough that in his early phase of career when he was a young doctor, he worked with Baruch Bloomberg at NIH, and he also contributed significantly in the discovery of HBV virus. And it is often said that had Harvey Alter stayed at NIH when he was working with Baruch Bloomberg, he would also have shared 1976 Nobel Prize with him. But okay, anyway, he has been awarded it this year. So the problem was that despite of the knowledge about HBV virus, and though they were excluding the patients who were positive for HBV virus from donating their blood, there were just unexpectedly 20% decline in the cases of post-blood transfusion hepatitis. 80% of people having blood transfusion were still reporting hepatitis cases. So this was again alarming, and this is where Harvey Alter contributed. So what he did is a very simple experiment. He took blood sample of 108 patients who had undergone open heart surgery followed by blood transfusion. So this cohort of patient is likely to catch hepatitis because they are undergoing blood transfusion, and out of them 12 patients indeed developed hepatitis. When they diagnosed for HBV, they found that only 4 of them were having hepatitis B virus infection. Rest 8 of them were not having HBV. They diagnosed for the presence of other viruses, for example, HIV and other known viruses at that time, for example, CMV, EPV, etc. But none of them were diagnosed in the blood sample of those 8 patients. So this is when they came up with this interesting name, which is non-A, non-B hepatitis. It somewhat solved the mystery of why still 80% of people are reporting hepatitis despite of having proper diagnostic test for hepatitis B virus infection, which is an unknown agent at that time. NA-NBH virus is leading to rest of the hepatitis cases transmitted through blood. Harvey Alter's contribution is not limited up to here. He also helped in developing an animal model. So to carry out research, we cannot always experiment on human samples. In fact, we cannot experiment on human samples. That is not ethical. So it is important to have an animal model. Harvey Alter, he infected blood sample from NANBH infected patients into chimpanzees, and he found that chimpanzees also catch hepatitis infection. In fact, chimpanzees are the only non-human animals which can catch hepatitis infection. Some other most commonly used animal models in laboratory these days are mice and rabbits, which are rodent model. But at that time, rodents would not catch hepatitis infection until in 1990s approximately, some immunocompromised strains of mice were discovered. So this was again a very significant contribution in the field. But the identity of the virus remained elusive. People didn't know what is the nature of the virus. Development of animal model helped to some extent in some basic characterization. For example, the molecular composition or what is the size of the virus. But if this is a DNA-based virus or if this is an RNA-based virus, all this information was not known. This is where contribution of Michael Houghton is important. He is a UK-based scientist, born in University of East Anglia. Born in UK, and he went to University of East Anglia for a bachelor's degree in biological sciences, and he then pursued a PhD from King's College London. After which he traveled across continents and he joined a biotechnology firm in California known as Chiron Corporation. And this is where, along with his team, he contributed for this Nobel Prize-winning work. Currently, he is serving at University of Alberta, Canada. In fact, very interestingly, it has been very modest of Michael Houghton that he has always very vocally acknowledged his collaborators. He declined a very prestigious Canadian award, which was awarded to him, I think, in 2014 or 16, somewhere around that, only because of the reason it did not recognize the contribution of his collaborators. So this is the full team. He is Michael Houghton. He is Kim Lu Chu. He is George Kyo. And he is Daniel Bradley. Okay, so before I go on to discuss about what Michael Houghton and his team did, I would like to give you a brief concept about how immunity in our body works. So whenever there is any infection by any pathogen, which is a disease-causing microorganism, an army of immune cells identifies this infection and they go on to fight the infection. A specialized type of cell produces antibodies against some specific proteins on this pathogen, which are known as antigen. And it leads to the formation of antigen-antibody complex, which can then neutralize this pathogen. That is, it can render the pathogen unable to cause the infection or to propagate the infection. Okay, so now I would like to discuss the work of Michael Houghton. What he did is that he took blood sample of chimpanzees infected with NANBH virus, and he synthesized DNA fragments from this blood sample, which are able to encode for a particular protein or which are able to encode for the proteins. He then transferred the objective was to identify the viral DNA fragment or the viral genetic material from this infected chimpanzee, which is causing the infection. So he transferred these DNA fragments into bacteria, again by using a different type of virus, which is able to integrate a foreign DNA material into the DNA of a bacteria. What he then did is that he took blood sample of patients infected with NANBH virus, presuming that these patients would be having antibodies against the virus, and the bacteria which were infected. Among all these bacteria, the bacteria which was infected with the genetic material that has come from the virus will be having viral proteins. So when he reacted these colonies, these millions of colonies that he had transferred, that he had used to transfer this bacteria, when he reacted these colonies with the blood sample of patients infected with NANBH virus, they assayed for this antigen-antibody complex formation, and out of all the bacterial colonies that he screened, he found one clone that showed this antigen-antibody reaction. And in the words of Michael Houghton himself, he has said that we screened for thousands of millions of colonies, and among them we found just one clone that was positive for this antigen-antibody reaction. And it was literally like finding a needle in a haystack, because as in the very beginning slides, I compared the genetic material of human and virus. Approximately 3 billion nucleotides are present in a chimpanzee DNA, and HCV, on the contrary, is just having 9,000 nucleotides. So the objective was to identify these 9,000 nucleotides from a genome of 3.1 billion nucleotides. Okay, once they identified the bacterial clone, which is having viral proteins, subsequent experiments led to the identification of the virus itself. For example, they then identified that this NANBH virus is an RNA-based virus, which is having 9,000 nucleotides in its genome, coding for a single protein of approximately 3,000 amino acid, and so on and so forth. So this is when they coined this term hepatitis C virus and replaced NANBH hepatitis virus term. Now, the next challenge was, okay, animal model was developed, but if you want to actually design some drugs to test the efficiency and effectivity of a particular drug, it is good if we can synthesize virus in our own laboratory condition and not by using any animal model. So people at that time, that is the scientists working on the topic at that time, they were facing the problem that the virus won't multiply in cell culture conditions. This is where the contribution of Charles M. Rice are important. He is again a US based scientist. He did his BS in zoology from University of California, followed by a PhD from California Institute of Technology, after which he joined University of Washington, and this is where he conducted his prize-winning research. Currently, he is a professor at Rockefeller University in New York. So Charles M. Rice very carefully examined the genome or the genetic sequence of HCV virus, or genetic sequence of HCV, and he identified certain nucleotide sequences in this, you can say, head and tail region of the genome. When he incorporated these identified region in the head and tail region of this HCV genome, and when he transferred when he transferred this particle into some cells which are grown in laboratory condition, the virus multiplied. The cells then caught viral infection, and the virus multiplied inside these cells nicely. So once you achieve this, you can now actually synthesize an artificial virus, and you can then infect this artificially synthesized virus into any animal model that you want. For example, Charles M. Rice then infected this artificially synthesized virus in chimpanzee, and he showed that HCV alone can cause hepatitis infection. Now, owing to these research and these significant contribution from these people and others also, their teammates and some others which are not recognized in this Nobel Prize, we now have drugs available to treat hepatitis. You don't have to remember the structure and names of the drug, but some of the drugs, they act by inhibiting multiplication of viral genome. That is, they will prevent RNA duplication in the virus. And some of them will act by inhibiting synthesis of virus protein. Okay, so this is a little technical detail, but for those who are interested, what these drugs do is that the structure of these drugs is actually similar to some nucleotides which are present in our genetic material. So what these drug molecules do is that they will go and sit in the, for example, in the case of HCV, they will go and sit in the RNA molecule of the HCV. So when the next round of multiplication, that is when the virus will want to multiply its RNA molecule, when the next round of multiplication will occur, this is not a true nucleotide. So there will be addition of some mutations in that multiplication, or sometimes there will not be any multiplication at all. So this is how these kind of drugs work. And the drugs which act by inhibiting synthesis of viral protein, they are actually inhibitor of a viral protein, which is a protease, which is required for maturation of other viral proteins. Okay, so I would like to take this opportunity to briefly distinguish between drugs and vaccine, because I believe this is something which is not very well clear among us. A drug is something which I just showed. It is a chemical compound. It is something which is given after you catch the infection to fight the infection. That is, once you have the infection, you want to neutralize the infection, you want to make the pathogen inactive, so you will give this drug. A vaccine, on the other hand, is prepared by using the pathogen itself. So pathogen is a disease-causing organism. So a vaccine is prepared by using a pathogen itself, a less virulent, that is a less infectious or relatively weak version of pathogen is used to infect any animal model, such that your body will evoke an immune response against that pathogen. And those antibodies are used to prepare the vaccine, or for sometimes some specific proteins on the pathogen itself are used and they are infected in animal model to generate an immune response such that you will produce antibodies against that virus. And this is what is used for the synthesis of vaccine. A vaccine is given not after you have the infection, it is given before the infection, preferably in your childhood, such that your body is developing an immunity for that infection and you don't catch that infection in your lifetime at the first place. Fighting that infection will come later. You don't want to catch the infection. Okay, so the current challenges which are now coming back to HCV talk, this was just something which I wanted to share because I believe this is something which is quite confusing for many of us. So, okay, the current challenges for the field of hepatitis and fighting hepatitis is that there is still no vaccine for hepatitis C virus infection. There is vaccine for HIV and HPV, but HCV still is not having any vaccine. The treatment that is given usually by giving these drugs is not very affordable to everyone. A single dose of a drug is sometimes $1,000 expensive, and the full treatment will cost you approximately something in tens of lakhs if you want to go by drug-based treatment. And another major concern is that, as I mentioned in one of my previous slides also, HCV cases are very underreported simply because they won't show any symptom until late stage. So many people, they will be having HCV infection, but they won't know that they are having an HCV infection until it has transmitted into a very serious liver problem. Okay, so Ralph Bartenschläger has said he is a German virologist, who was also a contender for this year's Nobel Prize. And I would just like to mention his comment. He has said that I hope this year's Nobel Prize will bring attention to the silent epidemic of hepatitis C around the world. Towards the end, I would like to mention that in India also, there are some labs who are dedicatedly working on hepatitis C virus. One of them is Dr. Ranjit Kumar's lab in Indraprasth University, New Delhi. And another is Professor Soumitra Das's lab in Indian Institute of Science, Bangalore. There can be other labs also, but I could find at least about these two labs. Okay, so the take-home message from today's talk is a virus is an acellular structure composed of DNA or RNA, which is the genetic material, enclosed by a protein or lipid coat. Hepatitis is liver inflammation leading in severe cases, leading to liver cirrhosis or liver cancer. This year's Nobel Prize is given to Harvey J. Alter, Michael Houghton, and Charles M. Rice for their contribution in discovery of hepatitis C virus. Harvey J. Alter identified NANBH virus as another viral agent which can be transmitted through blood and which is causing hepatitis. And he also helped in developing the animal model. Michael Houghton, on the other hand, he carried out this screen using bacterial colonies and blood serum of NANBH infected patients. And he identified the molecular identity and nature of the NANBH virus and coined the term hepatitis C virus. Charles M. Rice did the most contemporary work. He identified some regions of nucleotide on the genome of HCV virus, which are required for the replication of virus in cell culture, which are required for the replication of virus. So once he identified that, we could now replicate the virus in laboratory cell culture conditions also. We now have drugs to treat hepatitis infection, but there is still no vaccine for HCV. Yeah.
Okay, thanks a lot. That was very informative, and I am sure that there will be some questions. So let me just take a look at the YouTube channel and see what questions are there. And I hope, Diksha, you will be able to help us answer some of them. So I'm just going to check on the YouTube channel. We have Surit, who wants to know what are the different methods used to detect viruses other than PCR and RT-PCR, because clearly people were detecting these viruses, you said, in the 1960s and 70s, where they did not have PCR. So how did they detect these viruses and specifically, say that, okay, this is this particular virus?
Yeah. So PCR test, they detect the genomic content of the virus. That is, they will detect if the viral RNA or DNA is present in a sample. In fact, for example, in this coronavirus only, other than PCR, we have rapid antibody based test, which detect if your body is having antibodies against a particular infection. And this kind of assays is what was used previously. At those time, people were detecting viral infection or some other infection for that matter based on antigen antibody interaction. They had some very simple assays. For example, in a plate, they will take a media and they will punch hole in that solid media. In one of the hole, they will put titer from a blood sample, and in another of the hole, they will put, so this will be the antigen, and in this will be the antibody, and in another sample, they will put the purified pathogen itself. So as I said, the antigen antibody interaction is very specific. If your sample is having the infection, it will be having antibodies against it, and you can then detect this antigen antibody interaction on this simple gelatinous solid material. So these were the kind of assays that were used in those time based on antigen antibody interaction and presence of antibodies in your blood against certain infections.
Okay. And there is another question which asks, like, why do these viruses infect specific species only and not all species? For example, you said that the hepatitis C virus infects, or at least initially, infects normally only chimpanzees and humans, but not other animals. So what is it that the virus is very specific to one particular kind of species?
Yeah. So to cause infection, viruses have to identify a specific moiety. And if I put it in the context of coronavirus, I think okay, a coronavirus, it identifies that this ACE receptor which is present on our lungs. So similarly, there will be some proteins which are able to bind this virus particle, which will be present only in some species. And these are the kind of proteins which are recognized by the virus to cause the infection. So these are some specific proteins which are not present in all the species, and which is why viruses have specificity in the infections that they cause, because of the proteins that they recognize to cause the infection.
Okay, so there is a question from Ram, who is asking that, how similar are these viruses? And if you know something about, one of them, can you use that knowledge to, find out about the others, in the sense that, for example, did we know about, did the knowledge of hepatitis B help us to figure out hepatitis C? Or, are these viruses all very, very different from each other?
Yeah, this is an interesting question, I think. So viruses, they are very versatile group of, I would say. So viruses are, I cannot say very versatile group of organism, because they are not true organism. So viruses are very versatile. They are having very different family. They are having different families, multiple different families, which are very different from each other. And in fact, there is how viruses evolved, that is also not very well clear, because some people say that some families of viruses, they evolved from some prokaryotes, and okay, they evolved from some bacteria like cell, and some family of viruses, they evolved from some animal like cell. And some people say that viruses are even older than any bacteria or any animal like cell. So okay, the thing is, these viruses are very much different from each other. And in terms of their structure, in terms of, as you will see, this is a typical structure of a HCV virus. It is having this spherical protein coat. And if you have seen structure of a coronavirus, it is actually known as coronavirus because it is having those spike proteins, which gives the appearance of a crown-like structure. Similarly, there are viruses which don't even have this kind of spherical structure. They are long, slender. They are having long, slender structure. This is about the structure, and their molecular composition is also different. As I mentioned previously, also some of them are having DNA as their genetic material, whereas some of them are having RNA as their genetic material. So viruses among themselves are very different, and this is unlike our human cell, or this is unlike any other kingdoms of life. For example, all the human cells, they will be having DNA only as their genetic material. In fact, all the eukaryotic cell, for that matter, eukaryotic as in all the animal or plant cells for that matter, and in fact all the bacterial cell will be having DNA as their genetic material, but viruses are not like that. And as far as the second question is concerned, did the knowledge about HBV helped in identification of HCV? Okay, so yeah, so what happened is that HBV, identification of HBV, only few years after the identification of HBV, there was a vaccine for HBV. But unlike that case, we still don't have any vaccine for HCV, even after 30 years of its identification. So I cannot say that identification or development of vaccine for HBV helped in the development of vaccine for HCV, for that matter. But yeah, it was what it helped is that it helped in the identification that HCV is also an infectious agent which is transmitted through blood, just like HBV. Other than that, I don't think there was any similarity between HBV and HCV uh, that helped in the work that I just described. These are two very different viruses belonging to very different families of viruses. They are only similar in the organ that they infect, that is liver, and in the mode in which they are transmitted, that is through blood and other body fluids.
Okay. All right. So actually, there are a bunch of related questions which I will take together. So both Kiran, Anushka, and Surit are asking about.
But just a minute,
Yeah,
I
go
would
ahead.
just add a point. When different diagnostic tests, not different, when diagnostic tests were developed for HPV, based on these immunological reaction, that actually helped for development of diagnostic tests for HCV and other viral infections also. So development of diagnostic test is something which is linked and which helped development of other diagnostic tests, because people only improved after that. They developed more sensitive and even more accurate diagnostic tests after that. But other than that, as far as their identification and vaccine development is concerned, I don't think they are related. Yeah. Okay. Sorry. You can go on to next question.
Okay. So yeah. Okay. Okay. Before I come to Harsh's question, yeah. So there is this one question. There are three sort of questions that's related. There is Surit is asking and Kiran is asking about this why do sometimes these viral symptoms happen only at a certain stage and not at the beginning? So you're saying that it happens after many years, and why are some cases of viral infections asymptomatic totally?
Yeah.
So this is the question about the timing.
Yeah, this is again a very interesting question. So there can be two reasons. Number one, for example, in case of hepatitis, what happens is that there is a possibility that virus is causing damage to your liver already, but you don't get a symptom until a very severe damage or a very big enough damage has occurred, such that your liver cannot such that your liver cannot get repaired after that damage. Then you can get symptoms. Or there is a possibility that a virus will have caused the infection. It will be there sitting quietly inside a cell without doing anything. This is known as dormancy or dormant period of a virus. And this is something which happens in cases of HIV, I think. You might be having HIV in your body, but you won't know it because it will be just there sitting quietly and doing nothing. And this is, I think, not very well clear why viruses have this dormancy period and why they stay quiet for some period of time and why they suddenly start manifesting their infections. But this is something which is probably the cause of why sometimes you have asymptomatic infections. And the other thing is that, for example, in hepatitis, as I just said, you won't be having symptoms until the disease has reached to its severe condition.
Okay. All right, wait one sec. Let me see. Okay, there is Anushka who's in the 6th standard and wants to know the difference between liver cirrhosis and fatty liver.
Okay, that's interesting.
And how is the liver connected to the other parts of the body? Oh, this is going to remind me of my biology book, something with the hepatic artery and the portal vein and all
Okay,
that.
I will go to my liver slide. You can see it here, but I will go. Okay. So fatty liver and liver cirrhosis. Liver cirrhosis is preceded by fatty liver. This is a kind of intermediate stage, and fatty liver is fatty liver stage is a reversible stage. What happens in fatty liver is that, for example, as I gave you example of alcohol consumption. One thing that alcohol consumption does is that because of the production or excess production of some of the byproducts that are released after alcohol metabolism, your body metabolism pathway shifts from breaking down of fatty acids to get energy into synthesis of fatty acid molecule. So this leads to enlargement of your liver because it is synthesizing more and more fats now, and this is morphologically visible if you take a liver section. I don't have picture here, but okay, this is morphologically visible if you take a liver biopsy section, and this is what is known as fatty liver, and it is fairly reversible. If you cut down your alcohol consumption, your metabolism pathway will again shift from fatty acid molecule synthesis to fatty acid molecule breakdown to generate energy. But if it persists for a longer period, it will lead to liver cirrhosis, which is as I explained here. Your immune cells will come, they will try to repair this abnormality which has occurred, and if again if again this persists for a longer time, these immune cells, they will keep on depositing some materials which are some proteins, collagen for example, and these materials, they lead to the formation of a kind of scar tissue, which is these lead to the formation and accumulation of dead cells which are not able to perform their function, and this renders liver unable to perform its function, and this is not reversible. This is not treatable, unfortunately, and people suffering from liver cirrhosis, if it worsens even further, they have to undergo liver transplantation. Otherwise, it can be fatal. And if the infection persists even further, it can lead to liver cancer. Was there any other question from Anushka?
Yes. Yeah, she said, how is the liver connected to other parts of the body?
Yeah. Okay. So location wise, as it is a part of a digestive system, I would say. It is situated right above the large intestine, and liver is having a duct which secretes its bile juices, which goes on to this gallbladder, and this duct, I think, something which is connecting liver to pancreas also, along with connecting liver to gallbladder, because a common duct runs from here which enters pancreas, both from gallbladder and liver. This is what I think, how it is connected to other organs. And then again, there are blood vessels which which connects every organ to heart. There are veins running, arteries and veins running from heart and carrying blood from one organ to another organ. So every organ is connected to heart that way, but I think liver is connected to other digestive organs.
Okay. Question: can you get hepatitis B and C at the same time? From Pradeep.
Yeah, that is possible actually, because in fact, in Harvey Alter's research, in Harvey Alter's research, he sometimes saw that some patients, this is not mentioned here, he sometimes saw that some patients were positive for both HBV and HCV. So yeah, I think this is possible. You can get both HBV, HCV together. It's just there is nothing exclusive mechanism that HBV will not cause HCV infection. Both the viruses can infect you at the same time. And if you are undergoing multiple blood transfusion, if you take blood from a patient who is infected with not patient, from a donor who is infected with HBV, and if for the second round of your blood transfusion you take blood from somebody who is infected with HCV, you can actually get both the viruses in your body.
Okay.
And this is what Charles M. Rice showed from his work, that he synthesized this artificial virus, artificial HCV virus, and when he infected this artificially synthesized HCV virus into chimpanzee. Chimpanzee caught hepatitis. So this showed that HCV alone is that is what at that time showed that HCV alone can also cause hepatitis. Other than that, there's a very well possibility of co-infection type of condition.
Okay, Pradeep has actually another question, which is, I'm not sure if I understand very well. Isn't hepatitis C virus RNA transcripted by hepatitis B DNA through evolution? Is that possible?
I think what it means is that has HCV evolved from HBV? This is what.
Okay, yeah, I guess that's the question. Okay.
Okay, so I would.
Do we know that or?
No, so actually, as I said, as far as I understand it, these two viruses are from different virus family. So I believe they would have they would have evolved differently in evolution. I don't think they evolved from each other. I don't think so.
Okay, and finally we come to the most sort of crucial question. Harsh asks, why is there still no vaccine for HCV, and what are the chances and challenges in getting a vaccine for HCV?
Okay, so as I have talked about it, okay, no, so that is not important here. So why any infection for that matter takes time to develop a vaccine is that by the time you know something about that pathogen, it mutates itself into another version of itself. I think you would have heard it in terms of coronavirus also. There are different strains of coronavirus which are causing infection. So similarly, there are different strains of a virus, and same is true for HIV also. There is still, I think, there's still no vaccine for HIV. Because by the time you get to know something, so how, as I described here, how a vaccine is developed is that you identify certain molecules on the pathogen which are able to evoke an immune response. And this is what virus and other pathogen tries to evade. They try their best to evade to escape immune response from your body. So if you identify a protein which is able to invoke immune response, by the time you go on to develop vaccine against it, the virus would have mutated itself into another type of protein which will escape your immune response, and that kind of infection will not cause immune response in your system. So this is the biggest challenge in developing vaccine against these kind of infection. And one of them is okay, underestimation I think is not important. Why there is no vaccine? Yeah, this is what I think. This is why I think there is still no vaccine, because of their high mutagenic capabilities. They mutate themselves very fast. And is there any possibility of developing vaccine against HCV? That was the next question, right? So yeah, there is hope. And as I would go on to this slide, as Ralph Bartenschlager has said, that this highlights the importance of HCV because it brings attention to this silent epidemic of hepatitis C infection around the world. So as we know, in every country, I don't know how many number of labs are working together to develop vaccine against coronavirus, and there is hope that we will get a vaccine by December, or maybe by January, or maybe by mid 2021 at least. So still, if you have labs working dedicatedly to develop vaccine against HCV, and because this year's Nobel Prize is awarded to HCV, it, I think, highlights the problem of hepatitis C infection globally. So I think hepatitis C will also get some attention, and I think more labs will work on development of HCV vaccine. So yeah, I think there can be vaccine. Let's be optimistic in that matter at least. Hello.
Sorry, I was on mute.
Yeah.
Okay, so there is a provocative question from Ram, who says he's heard of good bacteria and bad bacteria, but he's never heard of a good virus. And sort of follow-up thing to that is he's wondering if there are anything called good viruses, and if tomorrow magically all the viruses disappeared from the earth, would it sort of cause a problem? I mean, do they occupy some thing in this entire circle of life where they are playing some role, and if they disappear tomorrow, would it be good or would it not be good for humanity as a whole?
That's an interesting question. Same here. Even I have heard of good bacteria, bad bacteria, and I have also not heard of good viruses. I think there must be some, just that I'm not aware of them. I think there should be some viruses which are doing something good for us. Maybe preventing some infections in some species, but I am not aware of it properly. I will have to think about it. I think what will happen if all the viruses disappear tomorrow, all of a sudden. I'm sorry. I'm not very aware of good viruses.
Okay. All right. So maybe we take another question, which is, by the way, TIFR outreach is monitoring this. Are there questions on any other channels coming in? If they are, please unmute and ask them, because I cannot see it. I can only see the questions on YouTube. Okay. So there's a question that, of course, this is not on the this is not on hepatitis C, and I don't know if the question is also, how there's a question which says that, okay, somebody's responding to your comment on useful viruses, saying that there are bacteriophages which are useful at conveying that you can use them as some carriers.
So in this screen, in Michael Houghton's work, bacteriophages had very important role because he was able to transfer these DNA fragments into bacteria using bacteriophages only. So I completely forgot about it. So viruses which are able to infect.
There aren't any questions on the Facebook at least.
Okay. All right. So there is another question. This is of course not on the HCV, but there's a question that why is it that coronavirus, which is deadly, is not is asymptomatic or not? I don't think it's always asymptomatic. Who are in patients who are already infected with flu, and how come the immune response is robust against the flu virus, but not against the this SARS-CoV-2 coronavirus?
It's all about identification, I think. I will come to this slide. So here, how your immune system manifests an immune response is that it has to identify that there is some problem. And as I was answering to one of the question, why there is no vaccine yet. So your immune system has to identify that there is an infection. And what these viruses do very smartly is that they modify themselves such that your immune system will not recognize them. Your immune system will not recognize them at the first place and forget about manifesting an immune response after that. So this is what happens when, so what was the question? Why is there a robust immune response for common cold? So this is why there is no immune response for diseases like coronavirus, because immune system fails to recognize it at the first place. And I would like to say again about how vaccine work. This is what is utilized in development of a vaccine. You want your immune system to be aware of the infection. So for example, in cases of polio, what is given is that a weakened version of polio virus is infected in the child when he is given polio vaccine, such that your immune system will recognize it as something, so that it will manifest probably a weak response. And which is why whenever a child is given a vaccine, he is having mild symptoms. For example, he will have mild fever depending on the type of vaccine that he is given, such that when in the late stage of his life he is getting an infection. So now your immune system is able to recognize, okay, this is something which has caused some problem in the child or in childhood also. So immune system will have a memory. It will recognize it as something problematic, and then it will manifest immune response either by producing antibody or through some other means. For example, some cells are able to just simply go and eat up the pathogen, which is known as macrophagy. But okay, so this is what is important. Identify that there is something wrong is important, and this is what these smart viruses are able to escape. They don't let themselves get identified.
Okay. Do viruses evolve faster than other species?
Yeah, that's what I said. I think there are mixed opinion about that. Some people about at least this is what I think. I may be wrong in that. This is not my expertise area, but okay. So as far as I have understood it, viruses. Some people say that they evolved along with bacteria. When some bacteria they engulfed some other bacteria, and then they underwent a retrograde evolution. So in evolution, it is known that uh, you will evolve into a more complex species, but those bacteria then underwent a retrograde evolution, and they lost their complexity, and then they turned into something even much simpler, into something which is just a assembly of some biomolecules, and which obligatory requires another cell to multiply itself. So this is what some people think, whereas some people think that virus has evolved from, or virus is a kind of precursor from a eukaryotic cell, okay, for an animal cell, because there are some stretches of nucleotide sequence in an animal cell which represent some DNA based viruses. So some kind of nucleotide sequences are present in animal cell which are also present in some viruses. These kind of sequences are known as transposons, for example. So these kind of theories, it suggests that a virus was a precursor for an animal cell, and some theories suggest that it evolved from a bacteria. But and as I mentioned previously also, there are different families, very different very different from each other for viruses, and it is possible that different families have evolved from different kind of cells and at different time point in evolution. This is what I understand about viral evolution.
Okay. So I think that is sort of, are there any other questions on the other channels, people who are monitoring, or have we covered all of them? If not, perhaps Diksha, you can go to the last slide again, where we repeat the summary of the sessions coming up. Just to remind you, we are going to have Chai and Wai sessions every week. Next Sunday is going to be Diwali, probably. It's the Amavasya, and for a change, we will not have our usual fireworks session on Diwali, which we've had for all these years. But given it's Amavasya, it's a dark night, we will talk about the darkest thing in the universe, black holes, and which is part of the Nobel Prize in Physics this year. So an Amavasya is a good day to talk about black holes. So we will have that topic for a change.
I think we have covered everything. There are no questions on the Facebook.
Okay, so yeah, we will have, just a reminder that next week and we will have the Physics Nobel Prize Part 1. And following that, we will look at the CRISPR Prize in Chemistry and come back to the black hole at the center of the Milky Way at the end of the month. So do join us on Sunday morning at 11 o'clock for these sessions. And Diksha, thank you so much for taking us through the world of the hepatitis C virus and answering all the questions people have about it. Remember, if you still have questions, you can write to us at outreach, outreach at tifr.res.in, and we'll try and get back to you. So thanks a lot for watching, and be sure that you stay clear from take precautions and stay away from not just hepatitis C, but also the coronavirus that is going around. So, make sure that you wear a mask and keep distance and all that. Till we meet again, thank you and goodbye from Chai and Wai.
Thank you. Thank you all.